Good afternoon, everyone. Welcome to the Wednesday afternoon NIH Director's Lecture. A fascinating topic and a very highly qualified speaker to lead us through some interesting ideas about what's happening with immunology, the intestine, and health and disease. Fiona Powrie, who's here as our speaker, got her undergraduate degree at the University of Bath and then her PhD, or we should say DPhil, at the University of Oxford in immunology. After a postdoctoral fellowship at a company called DNAX Research Institute, she then went back to the UK and has been at Oxford pretty much for the last 20 years or so, moving through the ranks from fellow to senior research fellow to Sidney Truelove Professor of Gastroenterology to her current position as director of the Kennedy Institute of Rheumatology at the University of Oxford and principal investigator of the Translational Gastroenterology Unit, Nuffield Department of Medicine. She's a fellow of the Royal Society, member of EMBO, and has received the Louis Jeantet Prize for Medicine. And her interest in the intestinal immune system is something she's going to speak to us about. I also have learned that she's interested in educating public and scientists about what we all do in the laboratory and has played an interesting role in trying to promote that translational component to the public. Her topic today is entitled, in a provocative way, gut reactions. We in the United States are familiar with those at the moment. I will say nothing more than that. Gut reactions, host microbiome interactions in the intestine in health and disease. Please join me in welcoming Professor Fiona Powrie.
Well, thank you very much, Dr. Collins, for that very warm welcome. It's absolutely a pleasure to be back here at the NIH. I have many friends and colleagues. I've admired the immunology that has come out of the NIH for decades, and that's no change now. Very exciting work going on here, and it's wonderful to meet the early stage scientists. Always a treat on these types of visits. So I want to talk today about our interests in understanding host microbe interactions in the intestine, how they promote health, and how when they become deranged can lead to the development of disease. What I want to start with, though, is a message for early stage scientists in the audience. I know that this is not a specialty immunology audience, but one message for you is that this is a great time to be an immunologist, because what we are seeing is that immunity and deranged or deficient immune responses underlie a number of diseases that we hadn't necessarily associated with the immune system. So, of course, we know about the immune system in infectious disease. We know how it can respond aberrantly in autoimmune and chronic inflammatory disease, but we are learning that alterations in immunity and inflammation can underlie diseases such as neurodegeneration and mental health, type 2 diabetes, atherosclerosis, and others. So a great time with the tools that are emerging in our trade to be able to decipher the immune system in health and disease. Now, of course, chronic inflammatory diseases are complex, multifactorial diseases involving the interaction of genetic and environmental factors, often playing out through epigenetic changes that have an impact on the immune system as well as tissue responses. And when there is a maladaptation between the host and its genetic landscape and environmental drivers, that can lead to chronic inflammatory diseases. And in some individuals, they will manifest in the bowel with inflammatory bowel disease. In other individuals, inflammatory joint diseases, or in other individuals, inflammatory skin diseases. And of course, the challenge now as we think about how to treat these diseases is that there are shared components as well as unique, and the unique ones might relate to particular tissue-specific components. And of course, if we can understand and redefine disease based on processes that may relate to immune function in tissues, then we may be able to repurpose drugs across different diseases and move away to a new molecular taxonomy of disease, away from our siloed understandings based simply on medical clinical symptoms to more underlying process-driven taxonomy. Now, of course, an important factor in a number of chronic diseases is our resident microbes. We may consider ourselves a superorganism composed of our human tissues as well as our resident colonists, which of course include a vast array of bacteria, viruses, and fungi. And of course, they make up a large number of cells in the intestine. There are more bacteria in the intestine than anywhere else in the body. 100 trillion bacteria inhabit the intestine. And of course, they contribute through their genes, their proteins, as well as their metabolites, interacting with the host and impacting on nutrition, immune system development, and host defense. So a huge amount of interest, including economic interest, in how to be able to harness the microbiome, how it interacts with the host to promote health, and of course to understand when there is maladaptation of this interface, how this drives disease. So the disease we're particularly interested in is the inflammatory bowel diseases. Of course, they are a poster child, if you like, of these complex multifactorial diseases involving the interaction between a genetic landscape and environmental factors that plays out in this interface between the immune system and the resident microbiota. So inflammatory bowel disease is a complex range of clinical phenotypes encompassing Crohn's disease and ulcerative colitis. It has an increasing prevalence, particularly in Eastern populations. And probably the heterogeneous clinical nature is a consequence of the multifactorial interactions that are driving the disease. There's no cure for inflammatory bowel disease. A large proportion of patients will progress to surgery. Of course, there have been great advances in the last decades, and one of those is anti-TNF, pioneered in part by Mark Feldman and Tiny Maney at the Kennedy Institute for the Treatment of Rheumatoid Arthritis, but also applied in inflammatory bowel disease. And for some individuals, that has they will do extremely well, but a large proportion do not respond. And we don't understand who they are. We don't understand how to treat, what the course of disease is going to be. And of course, in this chronic debilitating disease with intestinal inflammation, there is an increased risk for colon cancer. Now, there is evidence of dysbiosis of bacterial communities in the intestine, and there have been a large number of studies done in this area. So if you look at the makeup of the different phyla that are present in the intestine, in a normal individual made up of Firmicutes as well as Bacteroidetes phyla, then in inflammatory bowel disease, this changes. There's an increase in proteobacteria, for example, and others. But of course, the question is whether this is a cause or consequence, and how we might use this information to be able to design therapeutic modalities. Of course, there's very good evidence from mouse models that when there is a genetic susceptibility giving you an IBD-like syndrome, and there are over 70 different models of colitis that develop as a consequence of genetic manipulation of the host, altering various types of pathways, but in very few situations do those genetic changes have an impact without a replicating intestinal microbiota. So this really is an interaction between the host genetic interface and the microbes. And of course, in IBD, some patients do benefit from probiotic therapies, and there are trials looking at fecal transplantation. So we have evolved with our intestinal microbes, and we're starting to understand scratching the surface, I would say, of the nature of those molecular interactions. So a number of the metabolites that are produced by our diverse array of microbes have a direct impact on the intestinal immune system. One, in promoting host defense at barrier surfaces. Two, metabolites such as short-chain fatty acids, which are fermentation products of certain types of bacteria, can impact on the types of immune cells. And of course, bacteria express a number of pattern recognition receptor ligands that stimulate different innate immune cells. So in health, of course, that is going to promote properly functioning dialogue and interface. Whereas when there is a host maladaptation, what we find is that some of these pathways start to drive chronic intestinal inflammation where the situation is maladapted. And that's really what I want to try to dissect today. So there are changes in the microbiome in inflammatory bowel disease. Is that cause or consequence? If we can understand them, can we reset the balance? But what about the genetic contribution? Well, of course, there's a very strong genetic contribution to inflammatory bowel disease. In very early onset inflammatory bowel disease, there have been a number of different mutations, over 60 different mutations that have been sequenced in monogenic diseases where the patients present with inflammatory bowel disease. They are differently penetrant. So for deficiencies in IL-10, for example, as far as we know, 100% of the patients who have a malfunctioning IL-10 receptor pathway will develop inflammatory bowel disease. But at the other end of the spectrum, the vast majority of IBD develops with a peak onset of around 20 years of age. And in that setting, genetics contributes for around 17% of the risk. There are a large number of SNPs that have been identified to confer risk, and I think there we might suggest that the genetics is giving a landscape that is permissive for a particular response to environmental drivers. But I think what the monogenic diseases have been highly informative in terms of identifying non-redundant pathways. And my colleague Holm Julig mapped these out, thinking about key aspects of intestinal homeostasis. And we can map these 60 different mutations into what we might consider as cornerstones of intestinal homeostasis. These are the ability to compartmentalize and exclude all of these resident microbes from the underlying lamina propria. So although they're beneficial, they have to be kept in their place, and we all know interactions like that. And that means having appropriate epithelial cell function, mucin secretion with a stout epithelial barrier that prevents the translocation of bacteria. The next layer of regulation, of course, is appropriate phagocytes that are able to take up any invading bacteria and kill them. And if there are deficiencies in some of these, such as in chronic granulomatous disease, for example, where a proportion of patients will develop inflammatory bowel disease, then that again is a maladaptation of a key aspect of intestinal homeostasis. Our own work has focused particularly on the role of regulatory T cells the so-called FOXP3 positive regulatory T cells that I'll explain in a little more detail in a minute. But a number of mutations in the FOXP3 regulatory T cell and IL-10 production are associated, will give rise to monogenic inflammatory bowel disease. So, of course, understanding the pathways attached to these monogenic diseases, through that we may be able to map those into larger pathways associated with the SNPs that relate to different processes that go wrong in different subsets of patients. So I'm suggesting to you that IBD is not Crohn's disease and ulcerative colitis, but rather syndromic, where there may be patients that have epithelial barrier defects versus phagocyte defects versus reductions in the regulatory, in immune regulation. So these are regulatory T cells. This is a specialized subset of T cells that develop in the thymus. They express the signature transcription factor FOXP3, and if you have a deficiency in FOXP3, then you don't have a functioning population of regulatory T cells and develop IBD and a range of other inflammatory diseases. So regulatory T cells develop in the thymus, they exit into the periphery where they control effector as well as adaptive as well as innate responses. Now, in addition to the thymic-derived regulatory T cells, naive T cells under certain conditions can be differentiated, can develop into FOXP3 expressing cells, and there's a lot of interest in understanding these induced versus more thymic-derived populations, what their specific functions are, and the division of labor in terms of controlling the inflammatory response. And of course, the gut is an important location for the differentiation of naive T cells into FOXP3 expressing cells. And of course, Wanjun Chen here showed the important role of TGF-beta in driving this process, and we, together with Yasmin Belkaid and others, implicated the vitamin A metabolite retinoic acid as a cofactor in driving this response. So certain environmental conditioning driving naive T cells into the regulatory into a regulatory T cell function. And when you're lacking this, it leads to a range of these different inflammatory diseases. So we have, over the last decades now, been studying the pathways that control intestinal homeostasis, and this is a mouse model that we described many years ago, and involves transfer of naive T cells into mice that have no T cells of their own, and that leads to a chronic inflammatory disease that is driven by the microbiota. If you transfer these cells into mice that do not have a replicating intestinal microbiome, then they don't develop disease. And this disease is characterized by an increase in a number of types of T cells, the so-called Th1 and Th17 response, with key cytokine drivers. And if you neutralize these pathways, you inhibit disease in this model. But you can also prevent disease, not only prevent it, but also cure it if you transfer the regulatory FOXP3 expressing population of T cells even into mice that have established disease. This is sufficient. These cells will home to the intestine where they proliferate, and in an IL-10 dependent fashion will resolve the intestinal inflammatory response. So a lot of interest in being able to manipulate the regulatory T cell axis in inflammatory bowel disease. So using this model, we identified a number of different cytokines and cell surface molecules that are involved in the function of regulatory T cells to be able to both prevent and cure established colitis. And of course, those pathways have now come out in those monogenic forms of disease. So the mouse models are highly simplified, and you're looking at an aspect of inflammatory bowel disease. But I think that that maps on to certain monogenic forms of disease that are also very severe. So often you will hear, oh, the mouse models, they're not predictive. Well, they don't represent all of the aspect of inflammatory bowel disease, and the challenge is to understand how to map different mouse models on to different populations, human subsets of inflammatory bowel disease. It is worth just noting that checkpoint blockade-induced colitis with anti-CTLA-4 is probably highly relevant to what was unraveled in these mouse models. So fast forwarding now to where we are, there's a lot of interest in understanding how different sorts of bacteria are able to select different subsets of T cells. So how particular types of Clostridia, for example, from Kenya Honda's work, will drive regulatory T cells. Dan Lippman defining SFB, a particular type of segmentus filamentous bacteria that promotes the Th17 axis, and how these local changes in the intestine may have systemic sequelae in various chronic inflammatory diseases. So the model that we have used over some years now is infection with the mouse pathogen Helicobacter hepaticus. Well, in fact, it's probably a pathobiont. It is a microaerophilic gram-negative bacteria, Helicobacter hepaticus, that colonizes the cecum and colon. It's interesting in where it lives. It lives in the gel mucus niche, where it establishes a lifelong infection, and in normal mice does not elicit immune pathology. But when there's a maladaptation in the host, and particularly when there is a deficiency in IL-10, then this can lead to immune pathology. So what we find is normal mice infected with Helicobacter hepaticus mount a robust regulatory T-cell response. And Dan Lippman has recently published a very nice paper tracking Helicobacter-specific T-cells, showing a very high frequency of Helicobacter-reactive regulatory T-cells in the adapted normal setting. But if we now make this maladapted by taking out the IL-10, by administering an anti-IL-10 receptor, then we see now that we get chronic intestinal inflammation, and that is highly dependent on the production of interleukin-23. So what we're finding is that certain sorts of maladaptation may have particular drivers of pathogenesis. And when we have a relevant microbial driver, here in the form of Helicobacter, with an IL-10 deficiency, that gives rise to a high amount of IL-23 and an IL-23-driven intestinal inflammation. This model is in fact anti-TNF refractory. So it doesn't respond as well to anti-TNF as it does to anti-IL-23, and that's something that I'll come back to. So over some years, we have put forward a model in which there is a crucial balance between the production of interleukin-10 and interleukin-23 that controls this response. So in the normal setting, Helicobacter induces a high amount of IL-10, and this might be an important mutualistic response. It doesn't induce immune pathology because it's driving a particular type of response from the host. Whereas when that response is absent, Helicobacter now drives a huge amount of interleukin-23 as well as interleukin-1 from monocytes and macrophages, and that drives the accumulation of these pathogenic Th17 cells and the development of chronic intestinal inflammation. So we want to understand the host and bacterial factors that control this balance. And of course, anti-IL-23 is now coming alongside anti-TNF in the clinic for the treatment of certainly psoriasis, and now there are studies looking at IL-23 blockade in inflammatory bowel disease. So I want to tell you about three new things today. One is bacterial mediators that promote the regulatory T-cell axis, a new innate inflammatory checkpoint, and then a pathway of anti-TNF therapy resistance that we think may be relevant to a subset of IBD patients. So I told you that Helicobacter hepaticus induces a strong regulatory T-cell response. How is it doing that? So Camille Dan in the lab, a talented microbiologist, asked the question whether Helicobacter makes anything that impacts on macrophages, which of course are sentinel cells in the intestine, that promotes the IL-10 axis. Helicobacter seems to drive IL-10 in vivo, and that has a physiological relevance. So what Camille did was to generate culture supernatants of Helicobacter and add this to bone marrow-derived macrophages and look at various functions, and she found that Helicobacter supernatant produced an activity that led to a high amount of IL-10 compared to the pro-inflammatory cytokine interleukin-6. And she went on to characterize this and presented evidence that this is a polysaccharide. It functions through signaling through TLR2. And what was different is that the Helicobacter polysaccharide activity functioned differently to pure TLR2 agonists, such as lipopeptides, PAM2 and PAM3. They were not able to induce this higher IL-10 to IL-6 ratio. So Camille went on to look at some of the downstream signaling components that are activated following Helicobacter polysaccharide stimulation versus LPS, a TLR4 agonist, or PAM3, a pure TLR2 lipopeptide agonist. And what she found was that the supernatant, this was a purified, enriched polysaccharide activity, preferentially stimulated a particular signaling pathway downstream of TLR2 involving a number of kinases and activating the Krebs transcription factor. And this pathway has previously been shown to link to induction of IL-10 and other negative anti-inflammatory regulatory pathways. So this signaling through TLR2 was differentially inducing a particular signaling cascade that PAM3 did not do, suggesting there could be another receptor involved here. RNA sequencing of the of differential, actually I think this may be a microarray analysis of differential gene expression from these macrophages stimulated with the polysaccharide or PAM3, the TLR2 ligand, showed also, in addition to the IL-10 versus IL-6 ratio, that this polysaccharide differentially induced the expression of a number of genes that were associated with a prominent Krebs binding signature. And that's a functionally specialized pathway in terms of aspects of anti-inflammatory molecules as well as tissue remodeling. So we think then that this is an example of a molecule produced by Helicobacter hepaticus that uncouples the inflammatory response from an anti-inflammatory and repair pathway. It does this through promoting Krebs phosphorylation and driving a number of downstream genes associated with a particular functional response that may relate to the mutualistic relationship with Helicobacter. Important outstanding questions is there another receptor here? And this may be a common pathway of commensal signaling through TLR2 that promotes differential gene expression. And of course, there are similarities then to polysaccharide A, identified by Sarkis Mazmanian and Dennis Kasper, that also signals through TLR2 and was shown to promote regulatory T cells. There is some thought there may be other receptors involved that together with this too, and that may be a common theme. And this may be a mechanism of mutualism. And of course, understanding the Krebs pathway in terms of intestinal homeostasis is the next step. So I want to turn now to a discussion of a new host pathway that modulates the inflammatory response to Helicobacter. Now this is a response not in a lymphocyte replete mouse, but in a particular strain of immune deficient mice that have RAG deficiency. And what we found some years ago is that the 129 strain of RAGs, by contrast with, for example, the black 6 RAG, following Helicobacter hepaticus infection, develops chronic intestinal inflammation. It involves many of the similar pathways that you see in a T-cell dependent response, so it is neutralized with anti-IL-23. But of course, there are no T cells in here, and that led us to further characterize the IL-23 responsive cell in this setting, which is an innate lymphoid cell. It expresses transcription factors related to Th17 cells, but it doesn't have a T cell receptor, but it has these cells are present in the intestine, express high amounts of the IL-23 receptor, and can respond to IL-23 promptly with the production of a number of inflammatory cytokines. Now, in the black 6 strain, this response does not lead to intestinal inflammation, but in the 129 strain, it leads to chronic colitis, which will ultimately progress to colorectal cancer. Now, if we deplete these innate lymphoid cells based on markers that are present on their surface, this inhibits the development of this disease. So there's a genetic susceptibility here to Helicobacter-driven innate colitis, and this was also a model for us to identify innate lymphoid cells, a new type of lymphoid cell present at barrier surfaces that can contribute to immune pathology. Of course, there have been a large number of studies characterizing these innate lymphoid cells. There are different subsets of innate lymphoid cells. I'm not going to get into all of these. What's very intriguing about these cells is that they're present in small numbers and they're able to amplify their function. So our work together with many other labs identified these subsets of innate lymphoid cells that can secrete cytokines, the ILC3s in response to IL-23, which can be host protective, but when they become deranged, can be immune pathological. So for some reason in this 129 strain, this response was leading to Helicobacter-driven immune pathology. So we wanted to map the genetic component that was involved, the genetic susceptibility to Helicobacter-driven innate colitis. So we did a classical back-crossing analysis. The 129 strain is susceptible, the black 6 strain is resistant, and we identified a region on chromosome 3 that we identified as the Hicks locus, and this was work of Olivier Boulard in the lab, that could protect the 129 when you had this bit of B6 from the development of Helicobacter-driven innate colitis. So the protective effect with the B6 allele is dominant. I'm not showing you all of this, we've published this. The functional response is contained within hematopoietic cells. And what is in this 1.7 or so megabase region of chromosome 3? Well, there are 9 genes and 5 microRNAs. And we had a look at what was in there, what sorts of cells were expressing these genes. And one of the candidate genes that we have become interested in, and of course I'm going to tell you a story about this, is this alpha kinase. And there are 17 non-synonymous SNPs in ALPK1 between 129 and B6 strain. But first of all, just looking at the characteristics using the congenic strains following Helicobacter infection, you see this very rapid mobilization of innate immunity, lots of neutrophils, monocytes, and eosinophils on the 129 background, and by contrast, a hugely stunted response on the congenic with that little bit of chromosome 3. So Grisha and Nathan West picked this project up from Olivier Boulard's original observation, and a transcriptomic analysis shows that the 129 has a very strong interferon gamma response gene effect very early on compared to the R17, the congenic, and you see this in terms of the production of interferon gamma mRNA. So we wanted to look at this candidate gene, alpha kinase 1, and this is from a family that have this alpha kinase domain, which have, really quite diverse functions throughout the body. Alpha kinase 1, there really wasn't very much information, some cell line work suggesting it's involved in actin in the cytoskeleton. A couple of recent papers relating ALPK1 to bacterial sensing, in fact in response to Helicobacter pylori and some other bacteria. This is primarily in cell lines, and what those papers showed is that ALPK1 plays an important role in promoting this particular innate signaling complex, the so-called typhosome, leading to induction of genes such as IL-8. So some host protective function of ALPK1 in epithelial cell lines from the previous literature. But we were thinking that ALPK1 may actually be a susceptibility gene for chronic intestinal inflammation. So in collaboration with the Sanger Institute, we produced a knockout of ALPK1 now on the black 6 background, so that would be resistant to colitis. But what we found is that ALPK1 deficient mouse on the black 6 background compared to the heterozygote develops a much more severe Helicobacter-driven innate inflammatory response. We crossed that onto the lymphocyte replete setting now to look at the adaptive model of colitis, where we have Helicobacter infection and neutralization of interleukin 10. And what we found in that setting is that when you have ALPK1 deficiency, together with Helicobacter infection and IL-10 blockade, you have a markedly worse colitis. This is based on colonoscopy score, but also histologically. And in fact, that associates with this very marked skewing towards Th1 cells. So when you have ALPK1 deficiency and Helicobacter infection with IL-10 blockade, so you've got your microbial driver and introducing one maladaptation, which is taking away IL-10, ALPK1 is also clearly an IL-10 independent regulator, because the disease is markedly worse with a significant skewing of the response towards a Th1 phenotype away from IL-17, which may, in some settings, have a host protective role. Now, ALPK1 mice given Helicobacter hepaticus do not develop overt colitis, but they are clearly not normal because they also have a very high level of Th1 cells following Helicobacter infection alone. So this is altering the nature of the microbial signaling, and then how that leads to the differentiation of T cells, favoring the Th1 response. So this effect of ALK1 is transferred with bone marrow cells, the same as we find with the congenic region. We think this is the relevant genetic lesion. So we ask the question, is there a cell intrinsic role in the function of ALK1 in bone marrow, in this situation, in bone marrow-derived macrophages, which we grow under particular culture conditions, and then they are stimulated with Helicobacter hepaticus. The red is the ALK1 deficient, the blue is the heterozygous control, and you can see at early time points that the ALK1 deficient macrophages produce higher amounts of inflammatory cytokines, particularly IL-12. There's a very strong skewing towards interleukin 12. So the Hicks locus then on chromosome 3 controls the innate colonic inflammatory response. Susceptible 129 strain activate a very early interferon gamma and interferon response genes with rapid recruitment of myeloid cells. ALPK1, we believe, is the relevant gene that is involved in this locus, and ALPK1 knockout mice show an enhanced Th1 response in both the innate and adaptive models of colitis. It functions in hematopoietic cells to control the inflammatory response and has an important cell-intrinsic role in macrophages. So when there is Helicobacter stimulation of the innate immune system, this doesn't lead to immune pathology because the response is controlled by ALPK1. When this is removed, this leads to a high amount, and particularly of IL-12, and subsequently then the Th1 response. And this is an independent checkpoint from the production of IL-10. So Helicobacter is doing a couple of things. One, it's activating a response that is normally controlled by ALK1, but it's also activating high amounts of interleukin 10. And if we take away these two adaptations, then now we have a chronic inflammatory response. So what is the role of ALK1 in priming versus effector function? And of course, how is this related to human autoimmune and inflammatory disorders? So I want to finish now with bringing this back round to subsets of human patients and how we can iterate between mouse models and the human disease. Of course, we have an increasingly well-stocked toolbox for being able to intervene in inflammatory bowel disease, anti-TNF, anti-IL-23 that I've mentioned to you, targeting the JAK-STAT pathway from John O'Shea's work. Lots of opportunities, but I think the major challenge is we don't know which ones to apply to which patients. So developing strategies to predict therapeutic outcome will be critical for delivery of individualized patient care. And of course, there is a big problem in IBD in terms of anti-TNF non-response. There is primary non-response and, of course, secondary non-response. And we really don't know which patients will develop that. And, of course, there are different possibilities. Is there a genetic predisposition? Is it just the state of disease? It's just a kinetic thing of when we come to treat them? Or are there actually different biological drivers? And that patients that are non-responsive to anti-TNF actually have a different process that is driving the disease. So Nathan West and Ahmed Hagatzi in the lab undertook an analysis of transcriptomic data sets from ulcerative colitis and Crohn's disease patients to identify cytokines that were highly elevated in both of those diseases. And they brought their list down to 6. Some of them we know a lot about, interleukin-1 and IL-6, so we didn't follow those. But actually top of the list was a cytokine called Oncostatin M. And this has been somewhat overlooked because it's not called interleukin. It's been around for a long time, but there hasn't been very much investigation of OSM in terms of gut immunity. So OSM is a member of the IL-6 family. It signals through a heterodimeric receptor of the common GP130 subunit as well as a unique OSM receptor, and it activates a number of different signaling pathways downstream of that receptor, particularly through JAK1 and JAK2. A lot of information about OSM in terms of its biology. It functions in the liver, in the acute phase response, aspects of hematopoiesis, the CNS, heart, cancer. It's been implicated across a range of different diseases, but not much known about the gut. So we've interrogated a number of different publicly available transcriptomic cohorts. This is the pediatric risk cohort looking at primary IBD, and what we found there in their data set is that OSM is one of the most highly differentially expressed in IBD versus control mucosa with a high statistical significance. So we went back to our own IBD cohort in Oxford to look at OSM and its receptor, and again you see a significant increase in OSM gene expression and its receptor in severely active IBD. So we wanted to ask the question, is there any impact of OSM in whether or not patients will respond to anti-TNF? So this is our discovery cohort from the ACT-1 phase 3 trial of an anti-TNF antibody. These patients are refractory to standard therapy. Colon mucosal biopsies are collected prior to treatment with anti-TNF, and then the patients are treated, and there's a histological analysis and endoscopic scoring. And we found that patients that were non-responsive to anti-TNF had high amounts of OSM, and in fact this was highly predictive in terms of specificity and sensitivity in identifying anti-TNF non-responders. This is another patient cohort here, a UC patient, and you see higher expression of OSM and its receptor in the refractory patients. And that response, although OSM is the highestly differentially expressed, it comes with a module of inflammatory mediators related to a number of chemokines and cytokines. And when there are patients that were module high, 90% of those were unresponsive to anti-TNF therapy. By contrast, the module low patients, the majority of those were anti-TNF responsive. So we've validated that across a number of different cohorts. This just shows you in our Oxford-based cohort that there is an increase in T cells and non-T cells that are producing OSM in IBD. So there seem to be a lot of different cell types that can produce OSM, including T cells and myeloid cells, but the cells that respond to OSM is highly restricted, and that appears to be primarily to mesenchymal fibroblast type cells and endothelial cells. So this is just gating cells from the human intestine into stromal cells or different components, and looking at OSM receptor expression, which is expressed on mesenchymal stromal cells as well as endothelial cells. And indeed, one sees that the stromal cells mount a very robust response to Oncostatin M with the expression of a number of downstream signaling pathway, which is distinct from IL-1 or IL-6, for example. Is there anything special about OSM receptor high fibroblasts? Again, looking in the human intestine, we can see these OSM receptor high or low cells, and if you gate on the OSM receptor high cells, then they are cells that express a number of markers of stromal activation, including certain integrins and the molecule GP38 or podoplanin. So if we look at OSM receptor high versus low cells, there are some things that are similarly expressed, but there is a particular subset of cytokines and adhesion molecules that are highly expressed in the OSM receptor high population in inflammatory bowel disease, suggesting that OSM is signaling through these stromal cells and creating a type of stromal activation that may contribute to disease with these inflammatory mediators. So finally, is there a role for Oncostatin M coming back to our mouse model that is TNF, anti-TNF resistant? Indeed, if we induce Helicobacter hepaticus with IL-10 blockade in the setting of OSM deficiency compared to wild-type mice, then there is an attenuated disease. What's relevant here is that that's not really in the induction and development of the disease, but more in the perpetuation. So we think that OSM is signaling its function is coming later on. So OSM then is a previously unrecognized mediator of intestinal inflammation and a potential biomarker of anti-TNF non-responsiveness. We believe it acts through activating stromal cells, constituting a novel signaling axis between leukocytes and fibroblasts. This may have relevance in a number of different diseases. It may be a key high-level driver of IBD chronicity and severity that is particularly relevant to anti-TNF refractory patients. So what we think is happening when there is a genetic landscape with a relevant microbial driver, there is a dysregulated inflammatory response, high amounts of interleukin-23, IL-1, TNF, but in some patients there's also a remodeling of the stroma that involves OSM, which might have an important role in sustaining the inflammatory response through a number of different chemokines and adhesion molecules that impact on what is recruited and what is retained in the inflammatory lesion, something we're looking at. So I want to finish here and come back to this concept of a process-driven taxonomy of disease, where we may be able to pull out pathways that are relevant across different diseases and to be able to treat the process as opposed to the clinical symptoms. So, of course, all of this work is the output and attributable to the wonderful postdocs and students who have come to my lab over the years. I've mentioned the individuals who contributed to this work. We have excellent collaboration with our colleagues in gastroenterology, Simon Travis, Satish Kashav, and others. In our studies on OSM, we had a very strong collaboration with Scott Plevy and colleagues at Janssen, who made available to us a number of clinical trials that had not been published and data that we could use towards understanding the OSM pathway, and of course the sponsors that support our work. So thank you very much.
Thanks for a fascinating presentation. We do have time for questions, and as usual, please approach the microphones, as people are already doing, so that those listening by video can hear. We'll start over here.
Fiona, a very interesting story. In the case of the TLR2 ligand from Helicobacter hepaticus that differed from the PAM3, since TLR2 forms two different heterodimers with TLR1 and TLR6, there could be two different signaling pathways for those two different heterodimers. Did you get a chance to look at whether that accounts for the difference in what you're seeing?
Yeah, we were able to use an antibody, actually, that pinned it down to one of those, and then the PAM3 is the relevant TLR2/6 heterodimer. So I don't think it's just the type of TLR2 makeup of the subunit structure. I think there's another receptor here. I think it's highly likely. Yeah.
Okay,
Which is why the lipopeptide is not giving the same signaling pathway as the whole bacteria.
So Fiona, this is a very fascinating story, and it would predict that in those cases where patients are TNF unresponsive, they would respond to blocking the Oncostatin M pathway. So are these trials going on? How do you block the Oncostatin M pathway?
Well, Janssen have an anti-OSM antibody, for example. So I think the thing then is to get it on the agenda, and of course IBD is a very busy space. So it's difficult to get new trials in IBD actually, because there's a high failure rate and because we can't predict the patients. But yes, I would be optimistic that eventually we will be able to move forward and test our hypothesis here, which is all we really have, in patients. I think it looks an interesting one to go for, and something that not many strategies out there are targeting the stromal compartment. I mean, it's not a novel concept, but to have a cytokine that sort of directly is involved in activating that gives a tool, perhaps, a way to intervene.
Really interesting. Regarding Oncostatin M, if I remember properly, it can signal through two different receptors, either GP130 with the Oncostatin M receptor or GP130 with the LIF receptor.
Yes, yeah.
And so what is the expression of the LIF receptor like on the intestinal cells as well in IBD?
Yeah, that was one of our reviewer questions. So we did go back to look at LIF on our stromal cells, and we don't see the LIF receptor there. Yeah.
Okay.
A lot of these immunologic factors have to go through the portal system into the liver. And there is new hypothesis developing in terms of the etiology of non-alcoholic fatty liver disease, in terms of cytokines and bacterial products. Do you have anything in your project that's overlapping with that, or no?
Sorry, I missed the first part. I got liver disease, but I didn't.
Well, when you have the bacterial products, immunologic things going on in the intestines,
so the portal
they go
vein.
through the portal vein.
Yeah,
and when
yeah.
they go through the portal vein, they eventually go to the liver.
Yeah.
And there's a new hypothesis, I think, in terms of in California, about this could be an etiology of non-alcoholic fatty liver disease, among the other hypothesis now offered. Do you have any correlation of that with what you're looking at?
No, we haven't done that. But if you think of something like primary sclerosing cholangitis, for example, where there's a liver phenotype with some IBD manifestations, I mean, there's a very strong gut-liver axis. And of course, in the NAFL type things, there is a microbiome that is supportive of that and a lot of interest in understanding the pathways. But we have not looked at the liver in this context.
Okay, thank you.
Yeah.
So this was a wonderful talk, and this is a very naive question. You mentioned that the peak of incidence of these inflammatory bowel diseases is by 20 or a little more, which follows by a few years for the involution, the start of the involution of the thymus. So that suggests that there might be something produced by the thymus that prevents the occurrence of the disease on the appropriate genetic or microbial background. So are those diseases more frequent in kids that have had their thymus removed for heart surgery when they were very young?
I haven't heard that. I'm not aware of that, but I don't know the specific data.
I don't either.
Interesting thought. Yeah.
Let me ask you one sort of futuristic question. So you're mapping out all these amazing pathways and players in this enterprise, but also pointing out how important it is in therapeutics to try to do this in a fashion that recognizes differences between individuals. So can one imagine, once we have the technology and the pathways a little bit better worked out, that one would personalize the intervention based upon every bit of data you could collect on the individual, and that might involve not just looking at circulating cytokines and immune cells, but actually in a biopsy, having a look to see who the actors are. And perhaps not even by just measuring cytokines, but doing as some of us are starting to do in other immune disorders, single-cell biology, where you look at all of the immune cells and find out which genes are on using RNA-seq of single cells. Maybe then try to figure out for that patient what are the actors that you most want to intervene with. Is that where you think this field may ultimately be going, to that level of specificity in terms of therapeutic selection?
Yeah, I mean, I think there's a lot of experimental work to getting get the human cell atlas and so on in health and disease, and this is starting to come. How this will play out into clinical practice, I think we probably need markers that are more accessible also in circulating cells. So a challenge will be to look for signatures and then see if we can detect them systemically. I think the thing we're missing at the moment in this is the microbiome component, because what we're finding in the mouse models is where there's a maladaptation, be it IL-10 or an autophagy deficiency or what have you, in that sort of set of things I set out, different microbes seem to interface with that. So that's another way of being able to stratify, actually. Yeah. Sure. And that might be more accessible as well.
It might,
yeah.
Especially the way things are going. Well, this has been a fascinating presentation that brings together a lot of cool immunology as well as learning about the intestinal tract. So please let us thank our speaker again and head to the reception.
Thank you.